Now we're going to start talking about the functional groups. The functional groups are the groups that attach to those carbon chains to give them their unique characteristics. So you're going to not only learn how to identify these functional groups, then you're going to put that information to use as you learn how to identify the four major categories of macromolecule. So these functional groups are going to give you the clues as to which type of macromolecule you're looking at. First time around, you're just going to learn how to visually recognize the functional group. And then you'll put that information to use as you learn to recognize the macromolecules. So first round, I'm not going to talk a whole lot about the macromolecules these are associated with. We're just going to go through the functional groups and you're just going to learn how they look visually. Okay, the first functional group we're going to look at is the hydroxyl group. And as I draw these on the board, I'm going to do something similar to what they've done here, but I'm just going to draw one carbon as a shortcut. Hydroxyl group is just this oxygen and hydrogen together, OH. Okay, so this is going to be our list of functional groups. Again, first round, you're just going to memorize what they look like. So the first one is hydroxyl. So I'm just going to draw a carbon. I'm not going to show anything else connected here. The hydroxyl group is the part in purple. That's the hydroxyl group. Okay, the second one is called carbonyl. And a carbonyl group is a carbon double bonded to the oxygen. It can be at the end of a molecule, in which case it will also have a hydrogen attached. Because remember, carbon needs to share 4 pairs of electrons, 1, 2, 3, 4. Or it can be in the middle of a molecule. And now carbon, 1, 2, 3, 4. Either way, it's a carbonyl group. So carbonyl group is going to be a carbon double bonded to an oxygen. So in this case, this whole thing is the carbonyl group. Okay, the third one is a combination of the first two in both name and in structure. So carboxyl. So the word is kind of a combination of carbonyl and hydroxyl, carboxyl. Let's look at a carboxyl group. This is a carboxyl group. It's always at the end of a molecule, and it is a combination of the two. So we've got the carbon double bonded to an oxygen, and then we've got the hydroxyl group. But when this all is happening together, this whole thing is called a carboxyl group. So of all the functional groups we're going to look at, these three look the most similar to each other. The next three are going to be very different from each other and different from the first three. I know this seems pretty dry right now. We're going to put it in context, and again, it's going to help you visually recognize the macromolecules. And these are the groups that give those macromolecules their unique characteristics, because again, otherwise it's just a carbon chain, and carbon is carbon. It's always going to behave the same. Okay, the next one is an amino group. In an amino group, anytime you hear the word amine, it's referring to nitrogen. So the amino group, number 4, is a nitrogen, and you'll sometimes see it with 2 hydrogens, and sometimes you'll see it with 3 hydrogens. If it has 3 hydrogens, it's the ionized form. 2 hydrogens is the non-ionized form. So amino is referring to this nitrogen right there. Okay, the next one is a very, very important one. Oh, I'm sorry, no. This one's important too, but the one coming after that is the really, really important. This is sulfhydryl. And it is just a sulfur and a hydrogen attached to something. You're going to see this in proteins. Okay, and then finally, usually phosphate comes before sulfide, I was surprised at the order of these slides. Phosphate is a very, very important group. This is going to be not only in DNA and RNA, but we're going to see this in ATP. This phosphate group, you've already actually seen a phosphate head on a phospholipid, and that's what this was referring to. Now this looks a little different because one of the oxygens has a double bond and the other three do not. You will never have to draw this, you will just have to be able to visually recognize it in Biology 102. So don't panic about that little detail there. If it has a phosphorus, it's a phosphate group. Okay, and I'm going to draw it down here. It's going to be a phosphorus, double bonded to one oxygen, and then the other oxygens are single bonds. So this whole thing is going to be a phosphate group. Okay, then finally, this last one, we won't talk about when we talk about the macromolecules. It is a functional group, so I present it to you now, but we won't discuss this one in detail until we get to DNA, much later in the semester. Okay, so this is a methyl group. And we know that we turn genes on and off by attaching methyl groups and removing methyl groups from the DNA. So methylated DNA is something we'll talk about when we get to that unit. But the methyl group is just a carbon with 3 hydrogens. And again, I'm not going to talk about that one in the context of the macromolecules. You won't talk about this one until we get to DNA. So when this is added to a molecule, this is a methyl group. Again, first time around seems pretty dry. We're just memorizing those functional groups. Okay, so now you know the different ways a carbon chain can vary. We know what isomers are, 3 categories of isomer. And now you know the functional groups. Now we're going to put all of that knowledge to work in understanding the macromolecules. Carbohydrates, lipids, proteins, and nucleic acids. We're going to start talking about what we call the macromolecules. Macro obviously implies that they're large. Some of these are large, and there are smaller versions of some of the larger ones. But before we understand macromolecules, we have to talk about 2 very important terms, monomer and polymer. You might have heard this term polymer in particular when we're referring to certain types of chemicals. Let's talk about what those terms really mean. Monomer and polymer. You can think of a monomer as really just being a unit, an individual unit, and then the polymer is going to be a chain of those units. You can almost picture them being beads. So if these were individual beads, these would be monomers, those separate individual units. And then if we link them together to form a chain of monomers, that would be a polymer. So polymer is really a chain of monomers. And in your body, you are constantly going back and forth between these 2. You probably mostly eat polymers. Most of you aren't eating individual amino acids. Most of you aren't eating glucose. So we're usually eating polymers and then breaking them back down into monomers, because monomers are what we need to get into the cell. Then in the cell, we can take these same monomers and rearrange them to form all kinds of different polymers again. So in the body, we're going back and forth between these 2 constantly. You'll see when we talk about the macromolecules that for 3 of those 4 categories, the monomers have some specific names and the polymers have specific names. And I'm going to draw a nice chart for you to keep track of that. This, of course, means reversible, and this isn't really a reaction. It's just showing you can really go either direction. But there are going to be 2 important reactions that cause this to happen. We're going to link monomers together through a reaction called dehydration reaction. And we're going to break them apart through a reaction called hydrolysis. Hydrolysis is really what you do when you're digesting your food. Let's look at both of these terms and think about what they mean before we even get into the details. Hydro. What does hydro refer to? Water. Lysis means to split or break, and this is water splitting. This is splitting using water. What we're going to do when we perform hydrolysis is we're going to add water to break these bonds. Dehydration. Do you think that's adding water or removing water? That's right. Dehydration reactions are going to remove water to link these together. But what do I mean by removing water? Well, looking at a little more detail at these monomers, if you can imagine that each of these monomers has a hydroxyl group on one end. Remember, hydroxyl group is OH, and then a hydrogen at the other end. So when we say that we're removing water, what we're going to do to link number 1 and 2 together in this chain is we're going to take a hydroxyl group from one and a hydrogen from the other. So dehydration reaction is going to be removal of water. We remove the water to link the monomers together to form this polymer. This polymer would still have a hydrogen on one end and a hydroxyl group on the other end. Because as we remove all of these, we're going to end up with a hydrogen left at one end and a hydroxyl group at the other end. So removing water to then link those two together. How do we go the other direction? We're going to add water back in. If we add water to break that bond with the help of enzymes, we're going to add an OH back to one end and an H to the other end, and you're going to go back to the way that looked before. So hydrolysis is going to be adding water. So adding water to break the polymer into monomers. And in dehydration, we're removing water to link monomers. Okay, to link monomers to form a polymer. So again, we're constantly going back and forth with these two reactions in the body. So this shows dehydration. In this example, they're just taking this fourth one and linking it up. The first three have already been linked. You can see that this already has a hydroxyl end. So we're going to take hydroxyl group from one, hydrogen from another, link them together. Removal of water, dehydration. That's how we're going to link monomers together to form polymers. And then hydrolysis. Adding water back in to break that back apart. Just taking it back to the way it looked before. So hydroxyl group goes to one end and hydrogen to the other. This term hydrolysis, hydrolysis is the name of the reaction. The verb would be hydrolyze. We would say that we hydrolyze these bonds when we perform hydrolysis. Okay, and then finally, we can have hydrolytic. And this is going to be an adjective. We can talk about hydrolytic enzymes. These would be the enzymes that help us break that bond. Hydrolytic enzymes would be in your digestive system. Hydrolytic enzymes are going to perform hydrolysis. So this would be the noun. Not to give you an English lesson here, but I just want you to be aware that we're going to use all of these terms. Hydrolytic, hydrolyze, and hydrolysis. So hydrolysis is the reaction. We're going to hydrolyze which is a verb, meaning split them apart. And we're going to do that using hydrolytic enzymes. So all those words are very closely related. Okay, now let's start looking at some specific examples and going through the details of the functional groups. We're going to start with the carbohydrates. But before I do that, I want to draw a nice chart for you to give you some general terms for the monomers and polymers we're going to look at related to each type of macromolecule. Okay, so I encourage you to draw this same chart. It's good to refer back to it to keep track of this information. Okay, so this is going to be our list of macromolecules. These are going to be the terms we use for the monomers for that macromolecule. And then these are going to be what we call the polymers. Okay, so starting with carbohydrates, our first category of macromolecules. The monomers of carbohydrates are going to be called monosaccharides. Means one sugar. The polymers are going to be called polysaccharides. And then we're going to have specific examples within each group. For example, the monosaccharides are going to be glucose, fructose, galactose, etc. The polysaccharides are going to be starch, cellulose. So we'll get to those specific examples, but just so you realize we're going to break this down even further as we go along. Okay, I'm going to skip lipids for now, because as you're going to see, they don't really follow this model. We're going to go to proteins. The monomers of proteins are called amino acids. And the polymers are called polypeptide or even a polypeptide chain. It's not called a protein because that chain is going to have to fold into a very specific three-dimensional shape to be considered a fully or a polypeptide chain. It's not called a protein because, as you'll see, a protein has a very specific three-dimensional shape, and this is just referring to the chain of amino acids. So a chain of amino acids is called a polypeptide chain. Okay, nucleic acids do follow this model. The monomers are called nucleotides. And then the polymers are called polynucleotides. Okay, so again, monomer is a very general term, meaning just the base units. Polymer then is going to be a chain of those monomers, also a very general term. Then within each category of macromolecule, we have some more general terms. So this is specifically describing monomers of carbohydrates. Monosaccharide is a monomer of carbohydrates. We're going to see specific examples of monosaccharides. Same for amino acids. We're going to see 20 different amino acids. Okay, and then finally, the last category of macromolecule, the lipids, they don't follow this nice model of monomers linking together to form polymers. So we're going to put not applicable. Lipids have some characteristics that unite them, but they're also very different from each other, and they do not follow this model of monomers linking together to form polymers. Okay, so this is the chart that we're going to refer back to every time we hit another macromolecule. And you're also going to learn to visually recognize them as we move through them. Okay, so we're going to start with carbohydrates as our first group. Then we're actually going to cover lipids next. And then proteins and nucleic acids really have to be covered together, and you'll see why.